the last months we had really intensive work, and we tried to strengthen the collaboration between the different tasks and PIs. And one example where we had good achievements during the last months, this is connectivity. So connectivity is organized on different spatial scales, and we can analyze connectivity using diffusion tensor imaging in the living human brain, also in patients. And this gives us an impression where the large fiber pathways are in the brain, and Jeff Margin is doing this job together with his colleagues at Saclay and Neurospin. But then we ask the question, are all the signals, is also information that we get true, or where are perhaps methodical artifacts? Where do we have to go into more detail in order to understand the connectivity? And what is the connectivity within the cerebral cortex? And this question can be answered on a microscopical level using polarized light imaging, which is developed in Jülich. So here we can really go to the micrometer scale. And together with Jeff Margin, we now bring together these two types of data in order to cross-validate what we have analyzed, and also to get a broader and more complete picture of connectivity in the human brain. However, since the connectivity is so complex in the human brain and we cannot address everything empirically, we also started to use modeling and simulation and try to understand what the signals that we detect using our polarized light imaging, what do they really mean. And here we have developed simulation based on the UQueen system in Jülich. This is one of the largest supercomputers worldwide, this is top 9, and we really scale up to the system and can address the question of how fibers are really located in this model and how this model reflects the reality that we have in tissues. So in my view, this is a good example for showing how the different disciplines and the different spatial scales and temporal scales also come together. And during these last months, we also learned that it is not enough just to do good empirical research, develop methods, but we really should develop methods to such a degree that at the end we have a result or a product. And this product can then be accessed by the scientific community outside. This is done through the so-called co-design projects to which we contribute. And specifically, we are addressing the Human Brain Atlas, where all the different data coming from the different fields, from the molecular scale, from the diffusion scale, from the functional segregation or cytoarchitectonic segregation, integrated. And we provide this atlas, hopefully in a few months, to the open science community. We also think that it's necessary not only to consider in this atlas human brain structure, which is complex, but also to take into account human brain function and cognition, cognitive behavior. So we are happy that through the open call, which just finished, we have now new partners with whom we can address this question, because it is clear that the structural functional organization and cognition behavior, these are different aspects that we have to keep in mind when addressing human brain organization and coming to a better understanding, including not only the normal human brain, but of course also the diseased brain.